Your body is made up of trillions of cells that are constantly dividing. The processes that make these divisions possible are mitosis and meiosis. Mitosis produces 2 identical body cells, while meiosis produces 4 gametes with slightly different genetic information. Without these processes, life on Earth would not exist the way that it does today. Let's explore these 2 cellular processes in further detail.
Mitosis is the division of the genetic material in the nucleus of a cell. It is used as a method of asexual reproduction, growth, and development. But mitosis occurs after the completion of interphase and is divided into 4 stages: prophase, metaphase, anaphase, and telophase. Before mitosis begins, the chromosomes, the structures that hold the genetic material, have already been duplicated and are loosely arranged inside the nucleus. During prophase, the chromosomes condense into tightly wound X-shaped structures. This is the first time the chromosomes can be clearly seen under a microscope. The chromosomes are now made up of 2 sister chromatids held together at the centromere. Prophase is also the first phase in which the mitotic spindle begins to form. The second phase of mitosis is metaphase, in which the chromosomes align themselves randomly along the center of the cell or the metaphase plate. Microtubules extend from the 2 mitotic spindles at the opposite sides of the cell and connect to each sister chromatid at a specialized protein site called the kinetochore, which is located at the centromere. Once each chromatid is attached to a microtubule, the cell enters anaphase. As the microtubules shorten, the sister chromatids are pulled apart to form 2 daughter chromosomes. The newly separated chromosomes are then pulled to the opposite sides of the cell, and telophase can begin. In telophase, 2 daughter nuclei form around each set of chromosomes. After these nuclei form, mitosis is complete. Immediately following mitosis is cytokinesis, or the division of the cytoplasm. By this point, the chromosomes have once again unraveled. In animal cells, cytokinesis occurs when a cleavage furrow is formed by a tightening ring of actin filaments. The furrow eventually deepens to the point where the cytoplasm is split in half and 2 daughter cells are created. In plant cells, components of the cell wall are brought to the cell plate between the nuclei by vesicles. Eventually, these components form a new cell wall between the nuclei, creating 2 daughter cells. The 2 cells created by mitosis are genetically identical to each other and the parent cell in both the information they carry and the fact that they are all diploid. While similar to mitosis, the process of meiosis has certain fundamental differences.
Now that you have learned about mitosis, let's explore the process of meiosis. Unlike mitosis, which occurs in body cells or somatic cells, meiotic cell division occurs only in the gametes. The process of meiosis has 2 cell divisions, however only 1 duplication of genetic material. Because of this, meiosis results in the production of 4 haploid cells as opposed to the 2 diploid cells formed by mitosis. So let's begin. The first phase of meiosis is prophase 1. During this phase, the nuclear envelope within the cell is broken down. Homologous chromosomes are paired up within the cell. Also, during prophase 1, crossing over occurs amongst homologous pairs of chromosomes. The process of crossing over occurs at a location on the chromosome centromere known as the chiasmata. The next phase of meiosis is metaphase 1. During this phase, homologous chromosome pairs are lined up on an axis in the center of the cell known as the metaphase plate. Microtubules are attached to each homologous pair. After metaphase 1 comes anaphase 1. During anaphase 1, homologous chromosomes are moved to opposite poles of the cell by the spindle apparatus. Telophase 1 and cytokinesis wrap up the first cell division of meiosis. After telophase and the first cytokinesis, 2 daughter cells are produced, each with duplicated copies of half of the parent cell's chromosomes. The first phase of the second division is prophase 2. In this phase, the spindle apparatus forms once again in both daughter cells produced by the first division. The homologous chromosomes begin to move towards the metaphase plate in the cell. During metaphase 2, the homologous chromosomes are lined up once again along the metaphase plate. However, this time the chromosomes are lined up individually, like in mitosis. Microtubules are attached to connectitures on either side of the chromosome centromere. In anaphase 2, proteins in the chromosome centromere are broken down so that the sister chromatids of the chromosomes can be pulled to opposite poles of the cell. The second division of meiosis is completed after telophase 2 and cytokinesis. 4 haploid, genetically varying daughter cells are produced. One important aspect of meiosis is its role in increasing genetic diversity within populations. Crossing over during prophase 1, as well as the independent assortment of homologous chromosomes, are both responsible for producing 4 genetically different gametes.
Some differences between these two processes include that mitosis results in 2 diploid daughter cells with a single division, while meiosis results in 4 haploid daughter cells with 2 divisions. The cells produced by mitosis are genetically identical because there is no crossing over. On the other hand, the cells produced by meiosis are genetically varied due to crossing over in prophase 1.
Mitosis and meiosis are the processes that make life on Earth as we know it possible.